Hello and welcome to your Biology Paper 2 for Separate Science 2023 paper walkthrough. Many different species can live together in the same habitat. What name is given to all of the organisms living in the same habitat? There's only one answer that you can give here, and it says tick one box, and that is community. I'm going to take this opportunity to tell you that if it tells you to tick one box and you tick multiple, even if you've ticked one of them correct, you will not receive that mark. If it says tick two boxes and you don't tick two boxes, you only tick one, you've just thrown marks away. Please, please, please make sure that you are paying attention to the instructions that they give you. Which species of birds in figure 1 that I have put over here do scientists think are most closely related? Tick one box. So if we have a look at their names, we've got the brambling, which its Latin binomial name is Fringilla montifringilla. The bullfinch, which is Pyrula pyrula. Chaffinch, which is Fringilla coelebs, and goldfinch, which is Cardulis cardulis. So which species in figure 1 do scientists think are most closely related? Well, if you have a look, you are looking for names that they've got in common, essentially. And if we look here, the brambling and the chaffinch have got that first part of the binomial name in common. Now, the binomial name is made up of the genus and species name of the organism. So the first part of the name is the genus, and the second part of the name is the species. In this case, both the brambling and the chaffinch are in the same genus, so therefore that's the box that we are going to tick. Scientists think the brambling and the bullfinch belong to different species. What evidence is used by scientists to classify the brambling and the bullfinch as different species? Again, ticking the one box. The brambling and the bullfinch are different sizes. Well, if you have a look around your classroom, you will notice that there's people of all different sizes, and we are all of the same species, so it's clearly not that. The brambling and the bullfinch cannot breed together to give fertile offspring. That is the correct answer. The definition of a species is where two individuals can breed successfully and produce a fertile offspring. So this is an offspring itself that can go on and have other young or other offspring. So that is the definition of a species. So if they are unable to do that, it means that they are not from the same species. 4 other species of birds, A, B, C and D, live in a habitat in the UK. Figure 2 shows how the numbers of each species of bird varied during 1 year. So we've got A, B, C and D on this graph. So we've got number of birds here and then the month there. It says use information from figure 2 to answer questions 0.1.4 and 0.1.2 to 0.1.6. So we'll have a look here. Describe what happens to the number of bird species of A, which is this line here, during the year. Okay, now if you have a look, it's for 3 marks and it's a describe. So what I would always do is look at the graph and go, right, am I being asked to use data? No, just information. So I don't need to quote any data. So now I'm going to look at the shape of the graph. If it's 3 marks, there's probably 3 different parts of the graph line. So if we have a look here, we've got one part of the graph line there, one part of the graph line here, and then another part of the graph line there. So that is where our 3 marks are going to come from. We need to mention what is happening within those time frames. So I would say here, if we have a look at it, you would say it has a constant number. So constant numbers, and that is from January to March. Okay. Then you would say from April to May, number increases. And then you would say it decreases from June to December. And those are your 3 marks for that one. So again, if you are given a graph and it is just describe, it's what I like to call say what you see, bit catchphrasey. All right, say what you see. If it's worth 3 marks, it's probably 3 plot points or 3 parts of the graph that you need to refer to. right? Or it might be 3, but it says describe, and then it might say use data from the graph. So if that's the case, make sure you use data, because that's where the other part's coming from, or the other mark point is coming from. They've not done that here. They've not asked you to use data, so therefore it's just describe those 3 points of the graph. In June and July, a disease affected the populations of some of the species. Which species had the lowest resistance to the disease? So what I would be looking at is I would first be highlighting on my graph the June and July section, which is here. And then what I would be doing is essentially tracking the graph lines. So I'd look at A, so we've got A up, and then we've got a bit of a slight decline there. Fine. B up, big drop. C up, slight drop, and D actually increases and just a slight drop. So for me, the one with the biggest drop there is clearly B. Okay, so that would be B. We tick that. One species migrates between the UK and other countries. Which species migrates between the UK and other countries? Give a reason for your answer. Well, if you have a look at that graph, there is one obvious species that doesn't essentially, if you look at it, it doesn't seem to exist in the UK for certain months of the year. And that is species D. So you can see here, it only occurs, it kind of starts during mid-May, and then by the end of September, it's all gone. Now, what that is saying to me is that it's present in the summer months only. Okay, so this is one of those birds that migrates south for the winter. So you do tend to see that, is that you won't get some birds appearing in the UK apart from during the summer months where they kind of return to breed or whatever, but that's what's going on here. This bird species is migrating, it's present the summer months, or you could say it's only present from May to September. You might say, and then there's words to the effect of, so you would say that it's not present in the winter months, or it's not present from January to mid-May, and then October, November and December. You could absolutely say that. You could say it's not present for all of the year, it's only present for some of the year, it's not present in winter. So there's loads of different ways that you can say words to the effect that I have there. A person's eyes can focus on objects at different distances. A person looks at a distant object, the person looks at a near object. The person's eyes make adjustments so that the near object forms a clear image. Which term describes the adjustment of focus from the distant object to the near object? And that is accommodation. Okay. Hyperopia and myopia are long and short-sighted. Adaptation is nothing to do with this, so it must be accommodation. Which structure in figure 3 that I've placed over here is where the image is focused. So you've got all these different parts of the eye there. And in this case, it's B, but while we're here, we might as well go through all the different parts of it. So we've got the retina here. This C part down here is the optic nerve. D is the cornea. Okay, A is what's called the sclera, which is kind of like the white part of your eye, kind of like the white bit that you see all the time. Your lens is there. This kind of gap here, that is your pupil. Your iris is the coloured part of your eye. And then here you've got your suspensory ligaments and ciliary muscles. So E here is your ciliary muscles. And then these little things here are your suspensory ligaments. Okay. You've also got kind of quite a lot of jelly stuff in your eye. If you've ever watched I'm a Celebrity and they're doing the fisheye thing, there's always that lovely pop and then all this juice comes running out. That is the jelly part of your eye, which is referred to this bit here. And there's actually a little bit here as well, and they're called humours. So you've got aqueous humour and vitreous humour. Just a little bit of a side there for you. So which structure in figure 3 is a muscle that contracts when focusing on a near object? Well, if you look at everything that we've just labelled there, the only thing there that is a muscle is E. What happens to the shape of the lens when you're focusing on a near object? So when you're focusing on a near object, what happens is your lens does this. Okay, so you focus on a near object, it's doing that, which means that it is becoming shorter and fatter, or more convex, more curved, more rounded, wider, thicker, fatter, any of those terms. I prefer becomes thicker. But again, any of the words that I've just mentioned there is entirely fine. And then when you focus on a distant object, it becomes much more thinner. Okay, so it's thicker and then thinner when you're focusing further. So focusing far away becomes quite thin, but when you're focusing up close, it becomes much thicker. The eyes can function in dimly lit areas and in brightly lit areas. The iris contains muscles. Describe how muscles in the iris help the person to see clearly when they're moving from a dimly lit area to a brightly lit area. So, for 2 marks, first thing I would refer to is the size of the pupil. So when you are in a dimly lit area, your pupil is really large because you're trying to get as much light into that retina so for you to be able to see as much as you possibly can. When you go into a brightly lit area, your pupil constricts and becomes much smaller because your body is trying to protect that light sensitive layer of retina. So the first thing I would mention is there would be a constriction of the pupil, or the pupil constricts. Right, now, if you've not written constriction of pupil, but you have mentioned reduction in the size of the pupil, or the hole or the gap has become smaller, that is absolutely fine as well. That's just a different way of saying it. But constriction of the pupil is the best biological way that you can say that. So you've got constriction of the pupil, okay, and that's because the muscles contract. Okay, so the muscles contract, and it's asking us describe how the muscles help. Okay, so we've talked about the muscles, so that's the second thing. So we've talked about the constriction of the pupil because the muscles contract. If you've not said that, you could say constriction of the pupils to reduce light entering the eye, or hitting the retina. Right, so those are 3 things that you can say. You only have to say 2 of those in order to get the mark. It is important to be able to react quickly. Many people think that drinking coffee decreases reaction time. Plan an investigation to test the effect of drinking coffee on reaction time. You should include the test for reaction time that you would use and how to make the investigation valid. Okay, so anything to do with valid, what it's asking you to talk about there are some controls. And then here, you're just asked to basically describe briefly what you would do to test the effect of drinking coffee. Now, if we're looking at drinking coffee, essentially what we are looking at is the effect of caffeine. Okay, so that's what you need to be talking about. So, first thing I would say is I would use the ruler drop. Use the ruler drop method. Okay, and then I would expect to describe it slightly. So holding ruler above the thumb. Okay, you've described it in some way, shape or form there. You may have chosen a different one. You may, for example, say use a colour changing screen on a computer, and then they have to either hit the screen if it's a touchscreen computer, or they have to press a key when it changes colour. Equally valid method. I'm just using that one because that's the one we've used in school. If you want to use any other ones talking about computers, then that is also absolutely fine. Okay, you would say that you would have to repeat, and then you need to calculate a mean. So repeat at least 3 times, calculate a mean. You need to talk about taking readings before and then after the coffee. Okay, and then you're going to compare the readings. Okay, now control variables. We are looking for same age. So we're looking for age, we're looking for biological sex, we're looking for maybe even BMI, and you might think, oh, hang on a minute, that's a bit weird. Why? Well, the larger you are or the skinnier you are, the caffeine will have different effects on you because it's to do with your metabolism, all right, and speeding up all that kind of thing. So, and speeding up your heart rate and stuff. So the bigger you are or the skinnier you are, that effect of caffeine will be different. Okay, you could also talk about levels of sleep. You could also talk about the time interval. You might have mentioned that up here, by the way. You might have said, you might have mentioned up here about the time interval, because you might say, do the test, holding the ruler above the thumb, repeat that 3 times, blah, blah, blah. Then you would say, drink a certain, you might have said, drink a 100 ml coffee, wait 15 minutes, and then repeat the test. Absolutely fine. You could have written it up there. Again, that would be a great control, and you wouldn't actually have to mention any other controls because you've mentioned some there. All right, so that's what you're looking for. Now, in order to get your 4 marks, you have to reference collecting results with and without coffee. So if you've not mentioned there, and you've not mentioned any controls, you can't, even if your method is beautiful and you've mentioned repeats and all that kind of stuff, the maximum amount you can get is 2 marks. Okay, so you need to be wary of that. Reproduction can produce offspring which are genetically different or genetically identical. Farmers grow tomato plants in greenhouses and the tomatoes are sold in supermarkets. Suggest one advantage of growing tomato plants that are genetically different. All right, so we're looking for positives of variation within a population. So the first one for me, and the one that I would always go for first, is less susceptible to new diseases or pathogens. Now, you might not have used the word susceptible. You might have said means that they are less vulnerable. You might have described that by saying if a new disease or pathogen is discovered, or if a new disease or pathogen evolves, that means that these tomato plants are less likely to all die. So it's this idea of there is less likely for that species or strain of plant to die if they're all got genetic differences. You might also say that there is a variation in taste or colour. You can't just say variation, you have to name a characteristic that's really important. Or you could also say maintain the gene pool. Okay, now that's important because it means that we are going to reduce the chance of negative genes becoming more common within the population, which is what sometimes happens if you've got inbreeding or where you have them being genetically similar. Suggest one advantage of growing tomato plants that are genetically identical. Okay, well, they might all have, so instead of having that variation in taste and colour, they would all have the same taste and colour. You could mention that. Or they all have a high yield, okay, or same flavour, or same resistance to disease, or same resistance to drought. There's all sorts of things that you could say there. So they have the same named desirable characteristics. So high yield, same flavour, less susceptible to drought, less susceptible to disease, all that kind of stuff. Grow at the same rate is another thing you could say. And then linking on from that, able to harvest or ready for harvest at the same time, if they're all genetically identical. Scientists can grow genetically identical tomato plants using something called tissue culture. What is tissue culture? And that is where cells are grown into new organisms. So groups of cells grown into new organisms. Okay. Genetically identical tomato plants growing in the same garden do not all grow to the same height. Give one reason why. Well, there you've got to think about all the variations that can affect the growth of a plant. So anything to do with they have different amounts of water, nutrients, light. There could be different pH of the soil. There could be different plant densities, which means that they're kind of all crowded together and they don't have enough space. Different temperatures, different environmental conditions, not enough minerals or ions in the soil. They might have been predated upon by herbivores or insects or disease or pathogens. So there's loads of different reasons why an organism that is genetically identical might not actually grow to the same height or weight. The sex of dogs is determined by X and Y chromosomes in the same way as in humans. Now, hopefully, when Mr. Leake has spoken about this, he's told you that there are a number of different ways that they can ask this. Now, humans, it's XY. That's the only one that you're expected to know. Chromosomes are 46 in humans, 46 individual ones, 23 pairs in in body cells, and 23 individual chromosomes in our gametes or sex cells. Everything else in this world you are not expected to know. However, they all behave in the same way. So whatever number of body cell chromosomes you've got in the body cells, which is your diploid number, your haploid number will be in the gametes. Okay, and it just so happens that dogs have X and Y. Some birds don't. Some birds have birds have different ones. However, it will always be the same kind of process when it comes to using Punnett squares, where you have the female, which is going to be XY, and the male, which is XX. Now, some of you may not have even paid attention to the fact that it's female and male in that order. So if you wrote XX up here, or if you wrote XY there, you have lost that mark. So you get 1 mark for that and 1 mark for that. Okay, you then get 1 mark for this. Okay, so you then get 1 mark for putting all that together there. So that's what the 3 marks are for. A female dog gave birth to 6 offsprings. Why would you expect there to be 3 male offsprings and 3 female offsprings? Give your answer to this question here to help with your answer. Okay, and it's essentially that half are XX and half XY. You could also say that there is a 50% chance of either sex, or a 50% chance of male, 50% chance of female. Or you could say there is an equal probability of an X or Y sperm fertilizing the female's egg. Those are all different ways of saying the same thing. Farmers keep chickens for meat production and egg production. Some varieties of chicken grow more quickly and are more suitable for meat production. Other varieties of chicken produce more eggs. Farmer keeps 2 varieties of chicken, A and B, and the farmer investigated the growth rates and egg production rates of both varieties. So we've got growth, mean body increase in after 5 weeks in grams, and then the mean number of eggs laid per month. Suggest 2 control variables the farmer should have used in this investigation. Okay, well, clearly it needs to keep the temperature the same. Okay, because if you keep the temperature the same, that means that the chickens aren't wasting any of their energy maintaining their body temperature. The type or amount of food given, the light that they have access to, whether they're kept inside or outside. So if they are outdoor chickens, they're going to require more energy because they're moving around and they're maintaining their body temperature. So they're going to require more food, essentially, than ones that are kept in barns. So linking in with that, you could say the amount of movement or the amount of space or the particular time of year as well. All right, you could say the same stocking density, so basically making sure that you've got the same number of animals. You could talk about the same number of each type of organisms as well. So you can't just say make them have the same number, you need to say what that means. So it's the same number of each type of chicken. You could say the same starting mass. You could say the same age of chicken. You could say the same general health, or if they're on any underlying health concerns, or if they're on any medication, you wouldn't maybe include those. Figure 5 shows mean values from 500 chickens of each variety. Give the reason the farmer used a large number of chickens. All right, so every time we're talking about numbers, or we're talking about the number of individuals that have been chosen for a study, we always need to relate it back to the fact that it gives us a more valid or representative result. Okay, now I always get a little bit on my high horse whenever I watch these adverts on television that are saying like 99% of dogs or cats agree that Whiskers is the best one. And then you look at it and they've only asked about 20, or I'll say ask. They've only looked at the data for 20 or 100 individuals. That is not a representative result, really. Right, so the more that you have, the more representative it is. Also, it means that you've got less chance of inaccuracies or of anomalies, another one. So you might have mentioned reduced anomalies, or you might have said it's a more accurate mean, or more accurate results, or a more reliable mean, or more reliable results. Can calculate a valid mean is also another thing you could say. The farmer wants to produce a new variety of chickens that is good for both meat production and egg production. Describe how selective breeding of chicken varieties A and B can produce the new variety of chicken. So what we are looking at here is that you would, essentially it's asking how do you carry out selective breeding. All right, so there are 4 things that you must say, and if you don't say them, then you're in trouble. You have to refer to the varieties or the species or the strain or whatever it is. You have to refer to it at least once in your answer. If you don't, you're not going to get all 4 marks. So I would say breed the best of A and B together. Select offspring with, now here you have to mention what the desired characteristics are. You can't just say with the desired characteristics. You have to say with highest egg and heaviest mass or fastest growing. Okay, breed offspring together, repeat over many generations. Loads of pupils miss either, I'll show you where they miss. They miss either that, they miss what the desired characteristics are, or they miss this here, which is the repeat over many generations. So always check, have you mentioned what you are breeding together? Have you mentioned what the desired characteristics are that you are looking for? And have you mentioned repeat over many generations? Organic substances decay into simpler substances. The leaves fall off many trees in autumn, and the dead leaves contain carbon compounds and nitrogen compounds. Describe how carbon and nitrogen in compounds in the leaves are recycled and used by living trees. You should include a description of how the leaves are broken down and how substances are taken in and used by the trees. Okay. Now, when I was marking this originally, it was generally quite a poorly answered question. All right, you cannot get to level 2, which is your 4 to 6 marks, if you don't refer to both of these. If you just talk about how things are broken down, or you just talk about how carbon and nitrogen is used by plants, then you're only going to get a maximum of 3, depending on how well you've done that. So, first thing, I know that I have to talk about how leaves are broken down. So the first thing I'm going to talk about is decay. Okay, so decay caused by microorganisms. Now, if you want, you can talk about bacteria and fungi. You can talk about detrivores and all that kind of stuff. That's absolutely fine. So that's the first thing. So it's decay is caused by microorganisms. These microorganisms respire. Okay, and as they respire, that releases CO2. I wonder, how could CO2 be taken in and used by trees? That would be the next bit, wouldn't it? CO2 is taken in by trees through the process of photosynthesis. Okay, to make, I could say glucose or sugar or anything like that, that's fine. Now then, also decay also releases nitrate ions into the soil. You have to say that. You can't say releases nitrogen, they release nitrate ions into the soil. You then have to say that they're taken in through the roots, and that nitrates are used to build proteins. Loads of pupils didn't talk about the decay, or they said CO2 is taken in by photosynthesis, but that was it. They didn't mention nitrogen. If you're only going to mention carbon dioxide, you're going to get 1, maybe 2 at best if you say CO2 is taken in by photosynthesis to make glucose. Really, really important. Okay. Students investigate the effect of temperature on the decay of milk, and this is a method used. Place 25 centimetres cubed of fresh milk into each of 3 beakers, keeping 1 at 5 degrees, 1 at 15 and 1 at 25. Record the pH of the milk in each beaker every day for 4 days. So what we've got here is we have got the start, and then we've got 1, 2, 3, 4, and we're looking at the pH. So just one improvement the students could have made to this method. There are loads you could mention here. The one that I would go for is they've not actually mentioned the type of milk, so I would say same type of milk. In this case, I would be looking at same fat content, so I would be maybe saying make sure they use whole milk or they're using cream, right? You could mention that. Use more temperatures. Absolutely fine. Measure pH at smaller time intervals, so maybe every hour, maybe every 2 hours. Use a data logger for continuous monitoring. That's always a nice one. Measure pH at the same time each day. Repeat and calculate a mean, or repeat to eliminate anomalies. Cover or uncover all beakers. So those are things that you can say. If you've said anything else, that's not correct. It's only something that I've said there. So more temperatures, smaller time intervals, data logger for continuous monitoring, pH at the same time of day, same type of milk, whole milk, repeating, calculating a mean, or repeat to eliminate anomalies, and then cover and uncover all beakers. Right, here shows the results of 5 and 15 degrees on this graph. It then says complete figure 6, plotting the data from 25. So we are looking at this set of data here. Now yours is going to be more accurate than mine, but again, for 3 marks, you get 2 marks for accurate plotting and 1 for your line of best fit. Okay, so here we go. 0 is 6.8. Now, looking at these, it looks like for every 2 little squares, that is 0.1. So, for 6.8, I'm going to be up here. Okay, I'm going to be up there somewhere. Okay, then 5.3 at day 1. So, 5.123 is here. 2 is 4.9, so that would be here. Oh, no, it wouldn't. Tell a lie. Whoop. It would be there. Okay. Oh, point 1's gone there, hasn't it? So 5 point, let's go back to here, 5.3. No, not there at all. Not there at all. 5.3. Okay, then 3, it would be 4.8, 5, 6, 7. So again, remember that you're going to be doing this much better than I am. And then 4 is 4.8. Okay, which is in line with that. Now, the point 1's gone again. 5.3, about there. So you should have a curve that's freehand that looks better than mine, but has that shape. Okay, so that is what it is. Now, the line of best fit has to be a freehand curve. It is not a dot to dot. If you've done dot to dot, if you've done a straight line, you're silly because the other 2 aren't. Oh, well, technically kind of one of them is, but the other one isn't. All right, so it has to be freehand. The rate of pH change increases with an increase in temperature. The rate of pH change at 5 degrees is 0.3 pH units per day. Calculate how many times faster the rate of pH change is at 15 degrees C than the rate of pH change at 5 degrees at day 2. You should draw a tangent. Now, if you don't know how to draw a tangent, you're in trouble because it's going to happen a couple of times, all right? Particularly in biology, they like to do it for rates. All right, so ruler, I'm going to line it up with day 2, and I'm essentially going to try and draw myself a triangle to the best of my ability anyway. Draw a nice little triangle. There, like that. Okay, so... Oh, that was hard work. Right, so my y here and x there. Now, to work out the gradient of that line, it's change in y, which in this case is 1, divided by a change in x, which in this case is 2. So I'm going to go 1 divided by 2, which is 0.5. Okay, so fabulous. Nice little triangle there. Everybody's happy. Now, what I'm going to, so that is the gradient of my line there, which is 0.5. Now, if you have got that answer, you are bob on. Congratulations, well done. All right. Now, what we're going to look at now is how much faster this is compared to 0.3. So I'm going to go 0.5 divided by 0.3. And that gives me 1.67, and that is my answer. Okay, and that should also be your answer. All right. Milk contains lipids. Lipids are broken down when the milk decays. Explain why the pH changes more quickly when the temperature is higher. All right, so what we're looking at here is a little bit of your knowledge of how enzymes work. So enzymes are more active, or they work more quickly, or it is their optimum temperature. Oh, I'm trying to put in quickly. Enzymes more active or work quicker. What that means is that lipids are broken down faster. Okay, but how does that affect this pH change? Well, what happens when enzymes break down lipids? Well, lipase breaks down lipids into fatty acids. Fatty acids changes the pH, produced faster. Those are your 3 marks. If you do not mention this term here, you do not get that final mark. All right, really important. Homeostasis is the regulation of the body's internal conditions, and many internal conditions are controlled by hormones. Homeostasis works by negative feedback control. Figure 8 shows how the concentration of substance Q in the blood is controlled by negative feedback. So you've got an increase in the substance there. Concentration of substance is too high, so a hormone's released, it gets brought back down to normal. Decrease of substance, it's too low, a different hormone's released, and it brings it back to normal. Explain how the concentration of substance Q in the blood is controlled by negative feedback using information. So essentially, you are just describing what I have just said there. All right, so if Q is too high, A is released. If Q too low, B is released, bringing Q back to normal or ideal. Okay, because that's their ideal concentration. So you are just literally stating what I did there. So it's really important that you don't just ignore any diagrams that are there, because quite a few of you will have probably gone, don't need that diagram, and then explain, oh, I don't know what it's talking about. Whereas if you'd have understood that and gone through that figure first, then that question becomes really easy to answer. Thyroxine is a hormone produced by the thyroid gland. A decrease in body temperature causes an increase in thyroxine production. Explain how the production of thyroxine causes an increase in body temperature. If you do not know what thyroxine does, number 1, you're silly because you've not revised, and number 2, you've got no hope of answering this question. Thyroxine increases, trying to put 2 things in at once there. Thyroxine increases metabolic rate. How how does that link to increasing temperature, miss? Well, metabolic rate means that it increases metabolic reactions such as respiration. And what is the side effect of respiration? Releasing more energy. Okay, and therefore increasing the temperature. Now, you can say allows respiration releases heat, absolutely fine. If you said makes heat or makes energy or produces energy or creates energy, you're silly, all right, because energy can't be created or destroyed. You could also say respiration is exothermic, that is also fine. ADH is a hormone made by the pituitary gland. ADH controls how much water is reabsorbed from the kidney tubules. ADH stands for antidiuretic hormone. Essentially, if you are dehydrated, your body produces ADH, and what it does is it prevents your kidneys from filtering out the water into your bladder. So it increases the permeability of your tubules, making sure that more blood is reabsorbed back into the body. It's anti-diuretic. Diuretic essentially means anti-losing water in your urine. All right. Whereas when you are fully hydrated, you're not releasing a lot of ADH, which means that you are going to have lots of water being removed from your bloodstream and going and making urine. So let's have a look here. You can see here that as the concentration of ADH is increasing, the concentration of dissolved substances increases. So we've got an increase in concentration, and we have actually got a decrease in volume, which makes sense, because the less volume of urine you have, the more concentrated all of those substances are going to be. The concentration in a man's blood was 3.75, and the concentration of ADH in his blood decreased down to 1.25. Explain how the decrease in the concentration of ADH would cause the changes in the urine. All right, so what happens here is that when you have a less ADH, so less ADH means the kidney tubules are less permeable. So less water is reabsorbed. Okay, meaning that you get more water in urine, okay, which increases the volume, and as it increases the volume, it lowers the concentration. Okay, because essentially what you've got now is you've got a large volume of dilute urine. Okay, have to say all those four things. Okay, if you have said less water is taken back into the blood, that is fine. If you have said there's more water in the urine which causes an increased volume, that is also fine. Okay, there's different ways of saying it. You could say more water in urine causing increased rate of production of urine. Causing increased rate of production of urine. Loads of different ways you can say it, but that's what you need to say there. This question is about the effects of deforestation and agriculture. The process of photosynthesis and respiration affect the carbon dioxide concentration in the air. Figure 9 shows the changes in the carbon dioxide concentration in the air in a tropical rainforest during one day. The data are mean daily values over a whole year. So you've got during nighttime hours, you've got high concentration, then it starts to decrease. A peak of decrease is around about midday, and then after midday it starts to increase back again. Explain what causes the changes in the carbon dioxide from 0 to 6, and it's actually set it out for you, and 8 to 12. So you're going to get 2 marks for this and 2 marks for that. Okay, so really simple. All right, if you're between 0 to 6 hours, which is here, and then you've got 8 to 12, which is there. 0 to 6, it is dark. So plants respire. Really important that you remember that plants respire. Loads of pupils think that they only photosynthesize when they don't. So plants respire. Okay, if plants respire, they release lots of CO2. All right. From the hours of 8 to 12, it is light. So the plants photosynthesize. Okay. And what they do is they take in CO2. All right. Makes sense when you think about it logically. An area of rainforest is cut down and replaced with a field of maize. Only one crop of maize is grown each year. Figure 10 shows the changes in the CO2 concentration in the air in the field of maize during one day. The data are mean daily values over a 6-month growing period. Okay, so you can see there maize. Now, if we compare it to the rainforest, you can see that yes, there is a dip, but it's nowhere near as big a dip as the rainforest. The maize grows for only 6 months of the year. Explain why replacing rainforest with maize will increase the carbon dioxide concentration in the air after 1 year. All right, so number one only takes in, maize, I'm just going to put M because I've run out of space there, only takes in CO2 6 months of the year. How do you know that, miss? It tells you. Okay. And overall takes in less CO2 per day. Okay, because you can see that here. Okay, as well. So those are your two things that you need to say there. The values plotted in figure 11, which is down here, are percentages of the results of the control area. Why do scientists represent their results as percentages? And it's the same for the same reason, essentially, that you use percentage mass change in your osmosis practical. And it's for comparison, because not every area that you are going to study, not every forest area is going to contain the same number of trees. So that's the reason why. So it's for comparison, because we have different areas have different numbers of species, or different numbers of trees is another thing you could say. So trees or species. During the 100 years, the biodiversity of trees in the regenerating forest increases. So you can see this line here. So it's investigating the areas of rainforest that have been cut down at different times during the previous 100 years. So record the number of tree species, and we've got all that kind of stuff there. So this 100 there, that is the kind of like original trees. So or the original representation of what a healthy forest was. And you can see here, by the way, that it takes 50 years to get back to that kind of previous level once you've for regeneration, once it's been cut down. Anyway, during the 100 years, the biodiversity of trees in the regenerating rainforest increases. Give one conclusion you can make from figure 11. So that's something that I would go for. I would say that it takes 50 years to return to its original state, or it takes 50 years for the forest to recover. You could also say there's quite a few things you could say. So I would say it takes 50 years for the forest to recover. All right. You could alternatively say beyond 50 years there are actually more species than there were originally in the original forest. You could say that. Number of tree species, if you have a look here, increases rapidly at first, and then it increases slower later on. So that's another two things you could say. You could say the rate of increase of number of tree species decreases with time. So you can see that this curve of the line is becoming much more of a shallow gradient, which indicates that the rate is slowing down. You could say that there's a great variability of the number of tree species from one area to the next, and you can see that because you've got dots up here, you've got dots down here, so you see you've got a wide variability. So any one of those things is absolutely fine. Give two reasons why an increase in the diversity of trees in the rainforest leads to an increase in animal diversity. Well, all you've got to do is think about, well, what is it that trees provide animals? And they provide animals with food, and they provide animals with shelter. So we have more food availability, and we have more habitats. Or you could say more nest sites, more opportunity for shelter. With that more food availability, you need to say that there are different ones that are available as well. Okay, so not just more food availability, you have to mention that it's different sources of food as well. Okay, more habitats that are different. So if you have different habitats, that means that you are going to have an increase in biodiversity because you're going to have more animals there. Okay, so I'm just going to start over here. Hormones are important for regulating the menstrual cycle, and during the menstrual cycle, eggs mature inside follicles in the ovaries. Eggs maturing, by the way, is controlled by FSH. A 27-year-old female was infertile. Okay, so she has got an issue with the maturation or the release of eggs from her ovaries. A doctor tested a sample of the woman's blood. The test did not detect any follicle-stimulating hormone in the woman's blood. Therefore, the doctor has given the woman daily injections of FSH for 7 days. The doctor measured the concentration of FSH in the woman's blood, the concentration of estrogen in the woman's blood, and the volumes of developing follicles in the ovaries. Now, the follicles is the F part of FSH, and follicles are immature eggs, so we refer to them as follicles. So developing follicles are essentially maturing eggs. So they're not eggs yet because they've not been released from the ovaries, but they are maturing. So we've got this graph down here, and we've got start of injections at day 0. So we can see that her FSH is increasing. And then it kind of peaks around day 7, which is absolutely fine because that's when she stops, and then it drops off drastically. Now, as FSH is increasing, we can see that the volume of follicles that are developing is increasing, but then even though the FSH drops, it still continues, that volume still continues, which is quite interesting. Also, estrogen kind of also continues a little bit as well there. So give evidence from figure 12 that the follicles in the ovaries release estrogen. Okay, well, if you have a look, as the volume of follicles rises, the estrogen level is also rising exactly with it. If you have a look here, it's rising in line with it. So what that indicates to me, and it should do to you, is that as volume of follicles increases, so does the estrogen. Now, you might have said words such as correlation between, there is a positive correlation between estrogen concentration and volume of follicles, or estrogen concentration is in proportion to follicle volume. All right, that's absolutely fine. So as the volume of follicles increases, so does the estrogen. Next question. Injection of FSH causes the development of a number of follicles. The mean diameter of these follicles on day 11 was 22 millimeters. Calculate the number of follicles in the woman's ovary on day 11, assuming each follicle is a sphere, and it tells us how to work out the volume of a sphere there, which is 4/3 times pi times the radius cubed, because that's what r is. Okay, and it even tells you that here, what values to use or what they mean. Give your answer to the nearest whole number. Now that's important because you can't have part of an egg or part of a follicle because they are spheres. All right, so it needs to be to a nearest whole number. Now this is 5 marks. There's quite a lot of stuff going on here. So we need to look at day 11. All right, and we're going to look here at day 11. We're going to look at this over here, which is the total number of follicles. Okay, so we're going to go to day 11. Okay, which is kind of like here, and we're going to go all the way up to here, and we're going to see what the volume of follicles were. Okay, and that volume on day 11 is 39,000. Okay, so that's a total volume of follicles that was there. Okay, now what I'm going to do is, first off, I'm going to work out, so I've got this for later on, First thing now, well, second thing now, what I'm going to do is I am going to work out what the volume of one follicle is. Okay, and I'm going to do that because if my diameter of one follicle, the diameter of follicle was 22, that means that it's going to be, the radius is going to equal 11. All right, so that's the first thing we're going to do. So we're going to use this here, and we're going to go 4 divided by 3 times 3.14 times 11 cubed. And that is going to give me 5,572.4533, blah, 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 blah. Okay? Now, the total volume is this. So what I'm now going to do is I'm going to go 39,000 divided by the volume of one. So this is one follicle. So I'm then going to go 5572.4532, and that's going to come out with something like 6.99, blah, 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 blah. Whole number, therefore 7. Okay, easy as that, using this. Okay, now obviously a little bit of math skills, remembering that about 10 to 15% of your biology is going to be math skills. This is one of them, all right, and you've got 5 marks there. Right, nice and easy. That is the only answer there is. There is nothing else. Before treatment with FSH, the woman had underdeveloped breasts. Explain why the lack of FSH in the woman's blood caused underdeveloped breasts. Okay, well, you should know that lack of FSH also causes a lack of estrogen, because FSH triggers the release of estrogen. So no FSH equals lack of estrogen. Okay, and estrogen is the female hormone that controls the development of secondary sexual characteristics, and one of those is breast development. So controls development of secondary sexual characteristics. Oh, that should be a C there, i.e. breast development. Okay, so she's got no estrogen, her breasts are going to be underdeveloped. Right. Usually, males and females both produce FSH. You're like, what? Hang on, that's a bit weird. Now, if you think about it, it actually makes perfect sense. Because if half of a population didn't produce a hormone, pretty soon, none of the population would produce that hormone. All right, if you think about genetics and crossing and all that kind of stuff. So it's really important, actually, that both have the ability to make it. A woman has inherited a faulty gene for FSH production from each of her parents. Okay, so she's a faulty gene, right, for FSH production. Now, this means that she does not produce it. So she's therefore, female is going to be that, okay, is the female. Okay, the woman's parents both produce FSH. Show how the woman's parents could have a child that does not produce FSH. Now, if their child is homozygous for this condition, all right, where she needs one from each of her parents, so we know that she has to get the same one, all right, therefore we must know that her parents are carriers of this disorder, because both of them produce FSH. So both of them produce it, so they must have the correct gene, but they must also carry the faulty gene that she has got. So therefore, your Punnett square is going to look like this, H, 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 H. Then we're going to do the crosses. All right, now, loads of pupils leave it here and therefore miss the third mark. So you are going to get a mark for both of these being correct, and then a mark for that being correct. Okay, now what I'm going to, therefore your third mark is HH is 25% produces FSH. HH is a carrier, they are 50%, and HH is 25%, does not produce FSH. And you have to say that, you have to identify the genotypes, or you have to link the genotype with the phenotype, which is what does that actually mean. The woman continues to have injections of FSH, and the woman has a child with a man who is heterozygous for the FSH gene. Explain why the probability that the child will be able to produce FSH is 0.5%. Okay, so dad is heterozygous, so that means that he is this. The woman is this. Okay, so your answer is mother passes on H, okay, or passes on can only pass on the gene, which means that she does not produce FSH. Father has an equal probability, or is equally likely to pass on either a normal gene or the faulty gene, meaning there is a 50% chance the child will be HH, which is able to produce FSH, okay, or will be heterozygous. So that's what we're saying there. So those are your 3 marks. What can mum pass on? What can dad pass on? What does that mean for the child? Figure 13 shows 6 stages in the process of meiosis. You've got A, B, C, D, E, and F there, and you've got one little label that says 1 pair of chromosomes. In figure 13, A is the first stage, so we don't need to mention that, and F is the final stage. Right, and this is the process of meiosis. Okay, so it's a little bit different than mitosis, don't forget, because you get 2 splits. Okay, which is why you've got down at 2 splits and 4 cells at the end. All right, so remember, meiosis, 2 splits, 4 cells. And remember that meiosis is used only for the production of gametes. It does not produce anything else. So it only happens in the sex organs. Okay, so in females of humans, it's in the ovaries, and in male humans, it's in the testes. So in figure 13, the first stage, A and M, is the first stage, and F is the final stage. Stages B to E are not in the correct order. Give the correct order of stages A to F. So first thing is we get this doubling up of our chromosomes. We get this thickening there, so that is E. Then we get them lining up in the first division, which is D. Okay, and then being pulled apart to the opposite sides of the cell. We then get the kind of cell membranes reforming, kind of like that first division. So that's division number 1. All right, so that's going to go there. And then here with B, we have those chromosomes being pulled apart to other sides, opposite poles of the cell again, before you have that final split. So it's B there. Okay, so that is the only order that it can be in. So it goes E, D, C, B. At the end of meiosis, the number of chromosomes is different from the number of chromosomes at the start of meiosis. Give the number of chromosomes in one cell in figure 13. Okay, so figure 13, which is this here, okay, it says we're at the start. So this is the start here, all right, because we're seeing chromosomes and we've got pairs of chromosomes. So what I would be doing is going, right, number 1, I'm going to count how many pairs I've got. So I've got 1, 2, 3, 4, 5, 6, 7, 8. Okay, so that means I've got 8 pairs. Now, if I've got pairs, that means there's 2 of them. So therefore, I've got 16 individual chromosomes. If you have written 8 pairs, that is fine. Okay. Now, the end answer, therefore, is 8 individual chromosomes, because it's asking for the number of chromosomes. You would probably not get away with saying 4 pairs, because in gametes, they don't pair up. That's the point of fertilization. So just be wary of that. Explain why the change in the number of chromosomes is important. Okay, and we previously mentioned that meiosis is where gametes are formed. So it forms gametes that fuse at fertilization. Okay, now why is that important? Well, it means that offspring or embryos have the correct chromosome number. Okay, now you could say keeps the chromosome number the same from generation to generation, prevents the doubling or the increase in chromosome number. That's all fine as well. Meiosis produces cells that are genetically different. Describe how meiosis produces cells that are genetically different. So what you have is you have random chromosomes forming pairs. You also have or get moving from to one end of the cell, so you have that splitting, you have going into new cells or going into gametes. You may even mention the process of crossing over, so moves to one end of cell. Okay, so you might talk about crossing over, and that happens because chromosomes are a bit sticky, so when they all line up down the middle, you have those little like legs of the chromosomes, the chromatids. They might get stuck to each other, then as they get pulled apart, that genetic information goes with them. So that's a reason there. Chromosomes contain DNA. Figure 14 shows part of a DNA molecule. So we've got the all the different parts there. What type of substances are labelled A, C, G, and T? They are your bases. Okay. You can get organic or inorganic base if you want to, or you could just put base. All right. DNA is made of nucleotides. How many nucleotides are shown here? Well, you've got, split it down the middle, you've got 1, 2, 3, 4, 5, 6 there. You've got 1, 2, 3, 4, 5, 6 there. So you've got 12 individual nucleotides. And this here is known as complementary base pairing, where A always goes to T and G always goes to C. These little circles are known as phosphate groups. And these little pentagons are known as ribose or pentose sugars. Okay, and that's because remember that DNA stands for deoxyribonucleic acids. A wide variety of species exists on Earth. Most scientists accept Darwin's theory of evolution by natural selection as the explanation for this variety of species. Explain how our understanding of evolution has developed due to fossil evidence and increased understanding of the mechanisms of genetics. Now, you are not going to get into level 2 or 3. So level 1 is, well, level 0 is you've written nothing and it's just a load of waffle. Level 1 is 1 or 2 marks. Level 2 is 3 or 4 marks. And level 3 is 5 and 6 marks. Now, for level 2, you need to include a description about fossils and or genetics. But for level 3, it has to be detailed about fossils and the mechanisms of genetics. And you might be like, well, what's the difference there? Allow me to explain. So I would split this up into talking about fossil evidence first and then talking about genetics. So I would start by saying that what they do show evidence of. So they show evidence of life from the past. They show evidence of changes over time. They show the development of a species development over time as well. So it's a little bit of evidence for speciation as well. Also shows evidence for extinction. Okay, because there's no dinosaurs roaming around now, but we know they exist because we found their fossils. And it can show how organisms have developed or how they're exactly the same. So you've got like your crocodilians, like your crocodiles and your alligators and your caimans and all those kind of things that look pretty much the same as what they did during the time of the dinosaurs. Then you've got some that have changed massively over time. They've got a lot smaller. So fossil records show how organisms from the past are related to the species today. There are issues with this because there are gaps in that fossil record. These are being filled with new evidence when we find them. So the longer that humans have been here and the more that we dig up things, the more information that we are finding. The evolutionary trees originally were based on nothing but kind of physical relationships. So, for example, the giant panda and the red panda, the reason that the red panda is called a panda is because it's got a little thumb on the side of its hand, a bit like that, okay, which pandas have, which the giant pandas have as well, which means that they can both grip bamboo because that's the thing that they both like to eat. So originally scientists were like, they're closely related. Because of new genetic studies that we're going to talk about in a second, they've actually gone, actually, it's more closely related to a raccoon, which if you've seen a red panda, makes sense. Okay, so fossil evidence was how we originally made evolutionary trees because it was based on physical commonalities. So what do they look like? Genetics. Well, the father of modern genetics is Mendel, so you may want to mention Mendel and his pea plants, and how he came up with this idea of a factor of inheritance. His work wasn't very much, well, he tried to publish it in a few periodicals at the time, but they were like, no, because you're a monk, number one, and you're a mathematician, you've only looked at pea plants, a bit odd, and there was no evidence, there was nothing about DNA or genes at that time. He also came up with the idea of dominant versus recessive characteristics. So that's something else that you could mention there. Now, from that, the reason that we know that this actually happens is the proof that we have nowadays is our observation of meiosis, essentially. So it's observation of chromosome behavior. Because that previous question we were talking about, actually, well, previous question where it was looking at all the different cells, we actually had images of chromosomes. We know what happens inside cells now. We have visualized these chromosomes, we know what's happening, and we know what the mechanisms of inheritance are, and Mendel was actually right. So we actually observe chromosomes during cell division, and they have actually corroborated or agreed with what Mendel said. We also now understand the structure of DNA. That last question. All right, looking at, we know what the bases are, we know about complementary base pairing, so that all works now. We've also used this to understand evolution because there are variations in species that are caused by genetics. And these variations in species, if you think back to what we know about natural selection, the reason that we get speciation is because there is a variation in species due to random mutations within a population. Okay, and individuals with advantageous characteristics, so advantageous characteristics, survive and breed. And then pass on their advantageous genetics to the next generation, and as such, you end up with new species evolving. You may also want to talk about the fact that we're seeing resistance to antibiotics developing in bacteria, and you can see that, and that's because they change they have a new generation once every 20 minutes. So that is why we are seeing such an increase in the rate of antibiotic resistance, and that new species arise when you have changes that occur to prevent reproduction and things like that. Okay, fab. So that's all the answer there. Make sure, in order to get 6 marks, you have mentioned the fossils and the genetics, and you've talked a bit about speciation. So for me, if you've just mentioned fossils and you've talked a little bit about how fossils are made, then you're probably going to get 2 marks. If you've mentioned the fossils and the genetics, and you've mentioned Mendel as well, then again, you'd probably get about 4 marks. But if then you start linking it to the understanding of evolution and speciation, that is where you're going to get your 6 marks.